All right, so then we're going to go on. We're going to talk more about water. That's where we left off last time. We were talking about water. And so water is polar, which is really important for its properties. When we talked about last time about how it has a very high heat of vaporization, and that's because it's polar. It makes very strong bonds. We get very strong bonds between water and water. You also get very strong bonds between water and other things that are polar. Okay, so sometimes we call water the universal solvent. And that's because it dissolves most polar things. It does not dissolve nonpolar things. Okay, why is water polar? So why is water? Go ahead.
Because of the uneven distribution of electrons.
Okay, so we have an uneven distribution of electrons in water. So if we look at water, if I was to draw it, I could draw it like this. Why are those electrons? So we have electrons that are in these bonds between the oxygens and the hydrogens, right? And so those are shared electrons. It's a covalent bond. Each atom contributes one electron to that bond. Why aren't they equally shared? Yeah, Lorenzo. Right, it's about electronegativity. So water is more electronegative than hydrogen. Oh, there's my periodic table. So if we look at our periodic table, we know that our electronegativity increases as we go over here, this direction, right? And as we go up, and oxygen's almost in the upper left-hand corner. We're not going to count the noble gases, but it's also, upper right-hand corner. And so you expect it to be very electronegative. In fact, if we're looking at Pauling electronegativities, the electronegativity for oxygen here, let me do that one in red, is 3.5. And the electronegativity for hydrogen is, anybody know it off the top of their head? What was that? I think it's 1 point, is it 1.4? I should have said it. I saw people nodding. I was blanking on it. Okay, so what we can do is we can subtract those electronegativities, right? If we subtracted those, we would get a 2.1, right? 2.1, which is a really big electronegativity difference there. And so what that means is, like I talked about last class, we end up with a really high partial negative charge on our oxygens, and we end up with a high partial net positive charge on our hydrogens. And really, it's to the extent that it's like half, and that's like half a charge, and that's like 2/4 charges. Okay, so a large fraction of a full charge. And so how would water dissolve? How would this help water dissolve things? So let's say we talk about sodium chloride. All right, so how's water going to dissolve sodium chloride here? Let me get the picture farther up. And essentially what happens is water is going to surround the chloride ions and the sodium ions. All right, and so water can surround those ions like what you see right here, where you have the positively charged side of water. So you've got the hydrogens are pointed at the negatively charged chloride ion. And for the sodium ion, you get the negatively, so you get the negatively charged oxygen pointing at the positively charged sodium ion. Okay, and you actually get shells and shells and shells. So you're going to get these waters surrounding, and then you'll get an effect, a slight charge effect that occurs after that, and you'll actually end up with a little bit of a shell around that as well. So water can make those sorts of bonds. Is this process going to be entropy driven or enthalpy driven? And why? Entropy driven, right? So do we see, so we go from something very ordered, right, to something much more disordered. So that tells me entropy, there's going to be a positive entropy change here. What about the enthalpy? If you ever dissolve salt water, does the solution get hot?
No.
No. So there's not a big enthalpy change there, right? It also doesn't get cold, right? That can happen too. Um, what's a reaction where you dissolve something in water and it gets hot? Does anybody know from experience? Did lab prep, maybe this has happened. We wouldn't like just regularly have you do this in lab. Have you ever hear about you're supposed to add acid to water instead of water to acid? You may not have heard that. But if you put acid in water or you put base in water, so sodium hydroxide or hydrochloric acid, and mix it with water, it gets really hot. In fact, in college, I wasn't being careful, and I had a graduated cylinder. I think I had acid in it, and then I went over to the sink to add the water to it, and it dissolved and got hot and boiled essentially at the beginning and shot acid all over me. I ended up okay. I didn't have scarred or anything. Washed my face right away. Had my goggles on. Okay. And a couple times in life where having those goggles on has been important. Um, okay, but that's what you can do in terms of reasoning these problems out. You know, you can tell if the thing doesn't get hot or doesn't get cold, there's not much of an enthalpy change. And certainly, we're going to have, we're going to have energy changes in biochemistry, but in terms of reactions that happen in cells, do you think they get really hot or really cold?
No.
No. Okay, that would be bad, right? Cells in your body are all supposed to stay at about 37 degrees Celsius. Okay. So any organism, well, not any organism, but any warm-blooded organism is going to want to maintain that temperature. Okay, so we've got water. Water can be polar. There are other molecules that can be polar as well. There are molecules that can be polar and nonpolar. What name do we give those? And I might use a slightly different name than you have heard in other classes. Call those amphipathic. So I'm going to say they're polar and nonpolar, but then I'm going to go back and refresh it. Have a polar side and a nonpolar side might be a way to think about it. Oops. And here's some examples. Okay. They have a polar side and a nonpolar side. Can anybody think of an example of one of these? What was that?
Amino acid.
Amino acids, yes, there are amino acids that do that. I'm not going to use them for my example, but yes, agreed. Soap, yeah, soap does that. That's how soap works. And soap is essentially fatty acids. Okay, so if I draw a fatty acid, excuse me, the structure for a fatty acid, it's going to look like this. It's going to be deprotonated. I love teaching biochem instead of gen chem because then I don't have to draw every darn atom. And you'll see I get lazier and lazier. My little zigzags for fatty acids get squiggly. It's like my signature. And so this side is polar, right? It's charged. It's got more charge here than it has back here. Does it have more charge here than it has there? Why not? I know I asked it in the way that you were supposed to shake your head. Do you know?
Resonance.
Resonance, right. It doesn't really exist like that. The charge is distributed across this. Okay? But still, you got more negative out here than you do over here. So we end up with a charged side. And you'll have to end up with some positive over there to compensate for it. Okay, and then this is going to be nonpolar. Does water like to be around? I'm going to personify water here. Does water like to be around nonpolar stuff?
No.
No. Do you know why? This might be your first introduction to really why. It's about entropy, okay? So if I'm a water molecule and these are my hydrogens, and I got my buddy over here I can hydrogen bond to, and I got my buddy here I can hydrogen bond to, and I got my buddy over here that I can hydrogen bond to, I can like flip around between all of them. I have choices, right? Entropy is about choices, about ways to do things. Are these okay? I forgot about those. The lab coat stack is going down enough that we don't worry about it. Let's say this is a non-polar molecule though. I'm water now, and I come up and I'm like, oh, I don't want to do this. So all I can do is kind of bond with my buddies over here, and I don't have choice. I have fewer choices because I can't really do this. So I've got to do this, and they've got to do this. So all of us next to this hydrophobic molecule we don't want to associate with, we have to sit here and we have to bond in certain ways, and we don't have as many possibilities. So it's a lower entropy situation. And so when you think about trying to minimize or trying to maximize entropy, right? If I was to take a container and let's say I just put oil in it. So now I'm going to make this oil. Okay, and then we'll make this water. Okay, I can put these two together. And mix them, right? And what's going to happen? I can mix them and then I can let them go, let it sit, and what'll happen? They'll separate, right? The oil will be on top. I got that wrong. I was about to get it wrong. Does it depend on what oil it is? I don't know of an oil that sinks to the bottom. There might be. There may be some hydrocarbons that are more dense than water, but I don't know of one that does that. So I could shake this though. What would happen right after I shook it? It'd be all separated, right? But then it would go back to this. Why does it do that? It does it because it's minimizing the contact between the oil and the water. So in this conformation, we have the least number of molecules of water having to touch the oil or sit next to the oil. And so what we've done is we have maximized entropy because all these other waters down here have all the choices. These ones can't bond so much. Does that make sense? And so we refer to that as water having ordered cages. So water forms, I'm going to call these low entropy ordered cages around hydrophobic molecules. Or parts of hydrophobic molecules, as is the case for our pictures below. Can you guys see these pictures okay in the back? Not too small? Okay, I was seeing a maybe, maybe not look. So let me see if I can make them just a little bigger. So what I've got here is I have 3 fatty acids, okay? For these 3 fatty acids, here we got that, here we got that. And can you see how the way they've drawn this, these waters are in a very ordered arrangement around those fatty acids. Okay, that's their picture of it. But this is how it exists. They're still going to be in motion, but they're going to have to make these ordered cages. So for these, when I put those water molecules around these 3 fatty acids separated out in solution, count these. How many water molecules are in the ordered cages here?
13.
You get 13 around 1? So 39. I remembered 13. I just counted 12. I'm going to trust your 13. Okay? So we've got 39 water molecules that have to take this ordered low entropy state. But when we go over here, we put these fatty acids next to each other. Now we only have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 molecules. And maybe they could have put a couple more in there. But it's less than 39, right? It's 14, maybe it's 16. So this is higher entropy because more of the waters get to get out here where they have more choices. Does that make sense? Okay, and this drives a lot of binding things in biochemistry. So if we think about why a drug might bind to a protein, usually there's a patch. It may have some hydrophilic bonding stuff, but it usually has some hydrophobic patch to it as well, if that makes sense. Okay, so there's the drug is going to bind. Maybe we've got some nice hydrogen bonding or an ionic interaction, but there's also a big hydrophobic patch as well. And if you can exclude the water from that hydrophobic patch, the water now has more choices. And so you've got an entropy-driven event. Doesn't mean the enthalpy isn't important, but entropy is more important. Questions on that? So if I talk about this sort of thing, I may ask you about this, and I want you to recognize, at least for now, be able to give an explanation for why or how this works. Talk about the ordered cages of water and how these sorts of bindings, how membranes form, right? That's the start of a membrane. I'm about to show you some more, how that is driven by entropy. Yes?
Would you say it's because there's less surface area?
It is because there's less surface area, but you have to tie it back to these ordered cages of water, okay? Okay, and so you can get all sorts of interesting structures that form with lipids to prevent having lots of interactions between water and the hydrophobic parts of those lipids. So here we have an example of a liposome, and here we have an example of a micelle, and in both these cases you have the hydrophobic tail sticking in on the inside. And what you can do, when I was in grad school, I don't know where this research ended up, if liposomes still worked out well. They were using these for drug delivery, where you take drugs and you'd stick them in here, and you'd get this to go in the bloodstream. They would travel around happily in the bloodstream, because remember, this is polar. And it'd go to a cell, and merge with the cell membrane, and it would dump its payload, essentially. Okay? You can also do, and I'm about to show you some stuff with an mRNA vaccine, where they've actually managed to get this sort of thing to work as well for drug delivery. So if you get an mRNA vaccine, like a COVID vaccine, which I'm not sure you're going to be actually eligible for this year, but if you get that, it's this sort of molecule that's used to deliver the mRNA. And then we're used to our bilayer sheets, right? If you've taken a biology course, which most of you have, and those of you who haven't maybe have seen this before, what we've got is, but we'll talk about it in more detail, we've got a bilayer here. So if we have a gigantic membrane, it's just like the liposome, except it's much, much bigger, right? Okay, it's so big, it's like sitting on, if you were like a little molecule sitting on this, it'd be like sitting on the surface of the earth, right? It would look flat to you. Okay, so let's go ahead. I said I'd talk about this. Let's talk about how these lipids, or how these amphipathic molecules are used in mRNA vaccines. So this is an example here. This is off, it looks like I pulled this out of some paper, but what they've got is they've got a series of these hydrophobic lipid molecules. I bet these other little guys, these are probably cholesterol or cholesterol-like, and we'll talk about that later in the course. And then, so you've got a mix of them. Some of them are on the inside too, and then you have your mRNAs. And what that does is, you probably don't have a feeling for this, but RNA is not very stable at all, okay? DNA is super stable, actually. RNA not so stable because it actually degrades itself, and we'll talk about that in a few weeks. But it will actually can degrade itself or help its own degradation. And so we've got this this thing here that has the lipids on the outside protecting it. Water interacts with the hydrophilic parts, and where it has choices, right? Because it can make a good bond to the hydrophilic parts. That's still a choice. But for the hydrophobic part, really can't, so it's just going to stay out here, bonding out here, and form those ordered cages. So no ordered cages here. This goes in. You get injected. That can happen. That's generally happening in like your muscle cells, right? In your muscle, it's around your muscle cells. You could pretend this is a muscle cell. I was actually before class trying to figure out whether these hit any specific immune cell. It was my impression that they didn't based on just a quick search. So what happens is all of your cells, this is a defense against cancer, all of your cells take things in, and when they, just the stuff they're making as they go, right? So as they're making things, they take some of their proteins, they break them down, and they display a part of it on the surface. And what that is that's for the immune system to recognize that something's wrong, right? Because what's happened over time, and by the way, your immune system didn't probably develop at all to prevent you from getting cancer, right? Because in the wild, we all had our kids at 14, and then we all died at 30, and hardly anybody gets cancer by then. Okay, cancer is disease of aging. So there are bacteria that can get inside your cells and infect things. Okay, tuberculosis gets in the lungs. You get that inside cells. All right, there are a number of different bacteria and viruses that will get inside cells and just stay there. And if you and they make things. And if all your cells are always displaying some of the things they're making, your immune system can catch that a lot of the time, right? I'm a cell and I'm always making stuff. So here's something I made. Okay, is it okay? Here's something I made. Is it okay? And if I put out something that's not okay, that's not me, presumably, my immune system's going to come and kill me. Okay. So your cells are all supposed to be working together and taking hits for the team when necessary. So this could be a muscle cell. What the muscle cell is going to do is it's going to take in that mRNA. The mRNA is going to be used by the cell itself, the regular cell machinery, to make, in this case, this is the spike protein from, I don't know if you remember, you guys maybe don't remember this at all, right? You guys were much younger when COVID was here, and maybe weren't thinking about molecules. But there's a protein on the outside. Whenever you see these pictures of COVID, it's like a spiky ball.
Yeah.
Okay, this is the spike protein that gets made. And what the cell does is it takes it and it's going to stick the spike protein on the outside, maybe. But this display cells is going to break the pieces of it and display pieces of it on the outside. And then your immune system presumably comes along and goes, oh, that's not supposed to be here. And it marshals the defenses to have the immune system ready if it sees something like this to be ready to go and kill it. Okay? So that's how and in general, that's how vaccines work, right? You show stuff to your immune system, and your immune system gets the troops ready in case it shows up. All right, that's a real low-level explanation. And those of you who had immunology know about how it works. Dr. Lasser teaches that course. It's a great course. Yes.
Okay, this might be a dumb question, but like a vaccine is just gonna help you if you get the disease. It doesn't prevent you from it. Because I know we
It
get the
does.
vaccine.
It doesn't prevent you from getting it. What happens is when you get exposed, you already have immune cells that are ready to take it out right away.
Okay,
So whether you want to call that prevent you from getting it or not, you still get the virus in you, and then your immune system comes in and it's just all set to go.
But how does that work with chickenpox? Because like people who get chickenpox, it stays with them their whole life. But then if you get the vaccine, you can still technically get it though, right?
Well, so vaccine. So I can't speak to chickenpox exactly, right? And it doesn't actually last your whole life. The vaccine doesn't. Because you ever hear shingles?
Yeah.
Okay, so shingles is chickenpox, but when you're older, so you need to get revaccinated when you're 50. So not all vaccines work 100%. Okay, so if some people can get the vaccine and still get chickenpox, that's what's going on. I don't know enough about the chickenpox to speak to it. I've not heard that, but certainly the flu vaccine, right? They do the flu vaccine every year. It's some level of successful depending on how well they've guessed what the flu is going to be. And then I don't know above and beyond that how well successful. And the flu vaccine immunity fades pretty quickly. And so does the COVID immunity, right? Probably most of you have had COVID 1, 2, 3, or 2, 3, 4 more times, right? So I've had it 3 times. It's gotten better each time. And that's because my immune system is better able to handle it each time. But it's not the COVID vaccine isn't so good that it actually hits us with that prevent thing, right? Doesn't take it out right away. At least, well, everybody's different with COVID. But for me, it doesn't take it out right away. But it's gotten more mild every time. Because my immune system's ready. It's seen it before. Questions? Okay. That was about the original concept we needed was amphipathic molecules, right? But they're useful in this mRNA vaccine, okay? And most of the vaccines are things that like grow in chicken eggs, okay? And so you get the chicken, and then you get the chicken eggs make the antigens, and you get injected with those, the antigens themselves, as opposed to your own cells making them. And that's what your immune system says. Oh, this is what this looks like. This is what flu looks like this year. Yes?
So the amphipathic molecules are used because it keeps it from like the thing from degrading itself.
It protects the mRNA. So you've got the mRNA in here. mRNA, if you were to just inject mRNA into your blood, a lot of it or all of it would get broken down by enzymes in your blood and all sorts of things in there.
So what is like the water doing? It's like holding that all together.
The water here is just on the outside.
Yeah.
So we have hydrophilic groups on the outside that help hold this in solution in your blood.
Okay, so it's because like the water is
right.
They don't want to move. The water doesn't want to move, so it stays together.
The water is moving fine out here. Around this, the water is moving fine.
Okay.
Right? No problem out there.
Okay.
But if you were to put water in here,
It would
where it's all hydrophobic, except where the mRNA is, the water would be high, it would be low entropy because it would have to be in a very ordered state. It would not have many choices.
Okay.
Does that make sense?
Yeah.
But in terms of the example or thinking about that, use this to think about it in terms of the ordered cages, not the mRNA vaccine, because that's more complex in terms of the picture. You can use this to think about, why these structures form is so that water doesn't have to interact with that, right? The water, we have more, when we get this very ordered structure, the water is much more disordered as a result. That's why we get it. Good questions. Anything else? I'm going to move on to, we'll talk about our basic intermolecular forces. Okay, let's do that. We're going to finish up with that. So intermolecular forces. All right, name me 4 intermolecular forces. Give me one.
London dispersion forces.
London dispersion forces. We might call them van der Waals. Depends on who you're talking about. If they're more physicsy or not. Dipole-dipole interactions. Yeah, Robin. Hydrogen bonding. What else? Robin? Because nobody else moved. Okay, go ahead.
Me?
Yeah.
Oh,
Ionic interactions. You can do an ion dipole. We can mix them.
Okay.
Well, let's talk about ionic interactions. Or ion-ion interactions. You guys did those in just the right order. I was planning on reordering them. But for my point, what can we say about the strength of these in the order they're written? They get stronger as we go down. Okay, so these are stronger. What else can we say about as we go from the top to the bottom that you use to explain 90% of chemistry?
More
Yeah,
charge.
Lorenzo?
They're more charged on the bottom.
Yeah, they're more charged as you go from the top to the bottom. So we increase our charge as we go from the top to the bottom. So let's talk about that then. You may not be used to thinking about London dispersion forces as charges, but they are. They're just very tiny charges. So let's describe those first. So London dispersion forces. These are temporary small charges, okay? I'm going to say instantaneous. I'm going to say induced small charges. So an example of this, let's say we had an argon atom, but this happens in anything, period, okay? So if I was to ask you, name all the types of charges for this molecule, molecule X, doesn't matter what X is, it could be water. Water has London dispersion charges as well, okay? It just has hydrogen bonds that are so much stronger that we never really worry about it. Does that make sense? It's good. It doesn't mean that all of these have all the ones below them, okay? But everything's going to have a London dispersion force that goes on. It just might be that it's just so small compared to whatever else is going on that we don't worry about it. But let's say we have an argon atom. All right, we have for argon, how many electrons do we have on the outside outer shell? 8, right? We're going to have 8 electrons in that outer shell. And so those 8 electrons are whipping around all the time, right? They're actually they're a cloud, but we're not going to treat them as a cloud for this kind of example, but they're a cloud, right? We could think about that, but we're going to think about them like they're actually in specific positions for the moment. And so we've got these electrons whipping around, and at some instant, this is absolutely over-exaggerated, you might have 5 of those electrons on one side and 3 of those electrons on another side, okay? Super exaggerated here, would not be that much. And if this argon atom snuggles up to another argon atom, let's say I have liquid argon, they have to snuggle up. What happens here? I've got these electrons here. What happens to the electrons in this argon? These push those over to the other side. There's there's partial negative charge here, partial positive charge here. It's going to push those electrons to the other side. And so then we'd end up with this sort of thing. So these electrons push. I'm just going to write that there to hopefully remind you if you're looking at the notes later. Okay, I'm not going to write the whole thing. So it's an instantaneous effect, though. That's one instant it looks like that. The next instant, it could be the opposite, right? The next instant, it could be everything's evenly distributed. So it's not permanent at all. And so what we have is tiny charges. This is super exaggerated. A tiny inhomogeneous distribution of charge here that causes an induced dipole over here that causes them to attract to each other. And this is why you can make liquid helium or liquid argon, right? This is why those things will stick together because of that. Now you can also get that happening in you get that happening in any molecule. So you can also get this effect in any molecule. The more electrons you have, the greater the summed effect, if that makes sense. Okay, it's additive. Okay, so it's not multiplicative or something like that. So if I was to give you two molecules, A and B, I'm purposely being vague here. How would you, let's say on the next quiz without saying anything from here on out. 3 in the pool, I said, I gave you two molecules, A and B. What would you use to figure out which one has more electrons? In desperation, because I hadn't taught you it, what would you do? Use the periodic table, yes. So two molecules.
Count individual molecules and count.
Okay, so you could add up how many total electrons they have, yeah? Right? In terms of by atom by atom. What number on the periodic table might you use for that? Well, I guess you could probably valence electrons is the best way to do it. But the more atoms it has, the more electrons it has, right? Or maybe even, or if you want a better proxy, the heavier it was, the more, that might be molecular weight. Depending on, how much time do I have? But the bigger one, okay? The bigger the molecule is, the more electrons it will have. And so that's why, if we had, we'll talk about fatty acids melting temperatures later, but that's why if I have a fatty acid that looks like this, see, I told you I was going to get lazy. Didn't take any time at all. Okay? But these two are going to stick together more than these two. Okay? Stronger London dispersion forces, or more, I should say. Why don't I say more rather than stronger? Because these have more electrons. Get more electrons doing this here. Okay? We have a nice repulsive force up at the top. Two negative charges. Does that make sense? Okay. And so we're doing biochemistry, so we're never really going to have anything that's like all hydrophobic. But when you think about gasoline or oils or anything like that, that's all hydrophobic. The bigger the molecule, the higher the boiling point, because there's more London dispersion forces. Questions? Okay. And so when you think of a big protein folding, we'll get to this. A big protein folding, it folds all over itself and has stuff on the inside. There's so many London dispersion forces going on the inside of that thing that it holds together really pretty well. Okay. 4 minutes, so we're going to move fast here. I really want to talk about the London dispersion forces because they're the most complicated. Let me define the other ones at least. Dipole-dipole interactions. Are these permanent or transitory? Permanent. They're actually permanent because we've got a distribution. We've got something where one atom's more electronegative than the other, so you've always got more charge over here. It might vary a bit, and it probably does vary a bit, just like the London dispersion forces, but that difference in charge is big enough that it's always present regardless of some electron movement. Okay, so we're going to talk about permanent, permanent partial charges on molecules. Okay, and I'm going to actually insert here, you can put a little caret in your notes, attraction between permanent partial charges on molecules. Okay, and you guys had formaldehyde today, right? Was that on the quiz? Okay, so formaldehyde, you end up with the formaldehyde, that's a ketone. Well, let's draw the hydrogens, otherwise it's going to look weird. So I've got a formaldehyde, and I've got another formaldehyde, and formaldehyde can line up like this because I have a partial negative charge here because oxygen is more electronegative than carbon, where I have a partial positive charge. And it's a permanent charge, so I have interaction between the partial positive there on that carbon and the partial negative here is the way you can think about it. Okay, dipole-dipole interaction. 2 minutes. Hydrogen bonding. I am just going to define in here, it's a little more complicated than this. Hydrogen bonds are just extra special strong versions of dipole-dipole interactions, okay? Don't tell a P chemist I said that. Okay, so they're extra special and they only form between what? Okay, right. So form between a hydrogen on a nitrogen, oxygen, or fluorine, and a nitrogen, oxygen, or fluorine. Okay? And this is biochemistry, so we're not going to really see any fluorines. If this is a drug class, we'd have a lot of fluorines in it. Like, if this is a farm chem, we would do that. Questions on that? So don't ever show me a hydrogen bond off a carbon or to a carbon. Okay? We're only going to have oxygens and nitrogens in here. And then ionic, you guys have that, right? I'll just write it down, but it's the attraction between full charges. Attraction or repulsion between full charges.